Hi, everyone. Welcome to our webinar. My name is Gabriele Colasurdo. I'm the Manager of Education and Patient Services at Myeloma Canada. Today's presentation is going to be on CAR T-cell therapy in multiple myeloma. This presentation was hosted by the Vancouver Myeloma Network, the local support group out in Vancouver, and they graciously offered to share this presentation with the rest of you in Canada. So here I am. I'm uploading it onto YouTube for everyone to be able to get access to this very important information about CAR T-cell therapy and how it works. Before we see the presentation, I guess I should just let you know on who the presenters are. We have two great presenters, Dr. Kevin Song and Dr. Kevin Hay. They're both hematologists at the BC Cancer Vancouver General Hospital, as well as professors at the University of British Columbia. And Dr. Hay, in particular, is a scientist at the Terry Fox Laboratory in Vancouver. So without further ado, I'll stop talking, and I hope you enjoy the presentation.
Great. First of all, thank you very much for inviting us to chat to you all about some of the recent updates in myeloma and some of the research we're doing, and in particular how this relates to this new therapy called CAR T cells and what that means. And yeah, as Kevin Song has said, really the best part of this probably will be the conversation afterwards, regarding all this. Kevin's apologizing for the lights. I might have to apologize for the sounds at my house. If you hear some, a 3-year-old crying, that's probably her trying to be put to bed. So I apologize in advance if you hear that in the background, but that'll be my challenges potentially. So really what we wanted to kind of focus on based upon what we were told to chat about was really about CAR T-cell therapy in myeloma and how it kind of fits in to the standard myeloma treatments and where we see it going in the future. So I'll do a little bit of background on what CAR T-cell therapy is and then hand it over to Kevin, who will continue on to try to fit where myeloma fits in with all those standard therapies in myeloma, and then come back at the end with some of the research that we're planning on doing. So, cancer is not just the cancer cells themselves. And, this is something from the National Institutes of Health in the US website, a very good website they have that's very general about, understanding cancer. But one of the things that they've really added to a lot of these understandings recently has been about how the immune system really interacts a lot with cancer. And so you see on the left there that, in a normal sort of state, cancer cells are actually recognized by the immune system and can be killed off by certain cells of the immune system. And so you can kind of have this attack phase that occurs. But at some point, when a cancer kind of grows up and really becomes more cancerous and is able to grow into the big masses and such that we see on scans, it's able to somehow evade killing by the immune cells. And here's a brief look on the right about how complicated a tumor actually is. So you see there's all these cancer cells that are the white cells in between here, these kind of whitish grayish cells. But even within that, there's all these different immune cells that get associated with this tumor. And actually, this is true with solid tumors, but this is also true in myeloma, in that there's a lot of intricate immune interactions that the myeloma cells have with the tumor. And, part of what the tumor does is it not only evades attacking by the immune system, but it can sometimes subvert the immune system to actually help it to support the tumor growth. So we've known for a while that there's this very interesting interaction between the immune system and cancer, and tried to think how we can harness this better. So cellular immunotherapy is this understanding that these cells of the immune system can be used as a therapy. And one of the main players in this is called the T cell. Now, the T cell is a part of the immune system that recognizes abnormal cells in our body, such as virus infected cells or early cancerous acting cells. But as I said, cancers have developed mechanisms to evade killing by these T cells. And so what we've done with chimeric antigen receptor or CAR T cell therapy is we put in this new modified receptor on the surface of the T cell that's called a CAR. And now it's able to recognize a target on the tumor cell. And this enables this T cell to, go back to its standard job, which is to kill abnormal looking cells. So it recognizes this tumor cell as being abnormal and will kill it. So how do we make these cells? Well, first, we have a patient who's in need of this therapy with cancer. We take the T cells out of the body through a blood draw that's a leukapheresis. For a lot of you who have had a autologous stem cell transplant, it's the same sort of procedure where you sit on the machine and get your cells taken off. And the T cells that we have from there are then engineered with this new CAR thing. And this is usually a gene that's put in via different mechanisms that can be used. But this new gene that's in these cells makes this CAR being put on the surface of the T cell. So now when we give it back to a patient, it's able to now recognize the cancer and kill it off. Now, it doesn't just come with the cells alone, but it's often accompanied here by some chemotherapy. And the reason for this chemotherapy in particular, it is a chemotherapy that helps prepare the body to receive these cells so that they can properly, respond in the body. So what does this kind of practically look like for a patient who receives this? Well, it starts with, first of all, even before this timeline that I show you here, with, identification of a proper, patient to be referred for it, et cetera. And then assessment of an actual time to remove the T cells from the body. And this is anywhere from 2 weeks to 4 weeks prior. Cells are removed, the CAR T cells are being produced like I demonstrated to you, and then they're brought back. And just before they get infused into the body, the chemotherapy is given, usually over about 3 days or so here. And then with these CAR T cells going in the body, what happens with this red line is this is the number, so to speak, of CAR T cells in the body. They start to take off and expand, because what's happening here is that this isn't just like a drug that you take off the shelf, right? And you give it at a set dose, and that's the dose, and it, peaks in the body soon after you give it, and it goes away. No, this is a living product, basically, because these are cells. And so once you've, put these cells into the body, they will actually, seek and destroy the tumor cells. And in the process of seeking and destroying the tumor cells, they get the signal back that says, hey, there's something here to kill, make more of ourselves, make an army. And so they mount an army. And that's essentially what happens within the first 2 weeks here, is they're mounting a large army of immune cells, CAR T cells like them, to really take over and kill off any of the tumor. And then once they've killed off a lot of the tumor, the army, the big standing army, so to speak, isn't needed any longer. And so it goes down, and they start to, die off and leave the body, these cells. And the hope is that in some cases, there's this longer, what we call persistence of these cells in the body. So they'll hang around for much longer and potentially provide a surveillance mechanism in the future to try to prevent the cancer from coming back. Now, the two main toxicities that are associated with this therapy really occur within the first 4 weeks, but it's mostly loaded within the first 2 to 3 weeks. And that's something called CRS and neurotoxicity. Now, CRS is cytokine release syndrome. And essentially what this is it's like a overactivation of the immune system. So you can kind of imagine that this immune system in somebody's body who's received these CAR T cells previously has been kind of sitting and not responding to the cancer. And then all of a sudden it's getting this supercharged signal to say, seek and destroy. And these supercharged signals that are going around the body, and these cytokines that are the way that the body immune cells use to communicate to each other, really triggers this massive reaction in the body. And it starts out by just having maybe fever and flu kind of like symptoms. So I don't know how many of you have had like a really bad viral infection where you have those shakes and chills. That's and high fevers. That's what CRS starts to feel like. But in some patients, unfortunately, it can progress where the blood pressure drops, they have difficulty breathing, and they might end up having to go into the ICU to get, blood pressure support or support for breathing. And occasionally people have died of cytokine release syndrome. You know, the good thing is that those people who make it through, it's entirely reversible so far as we can see. And and so although it is very scary, it is very short-term kind of toxicity. The neurotoxicity, it's kind of interesting because it can sometimes happen at the same time as the cytokine release syndrome, or it can happen after the cytokine release syndrome has resolved or gone away. And what this is, we're not quite sure exactly about all the reasons this happens. And this is a very active area of research. But we say just broadly neurotoxicity because it can be something as simple as somebody having a tremor, difficulty writing, difficulty remembering certain things. But it can also get so bad that there have been occasionally people who have had seizures or stroke-like symptoms with it. But again, like cytokine release syndrome, in the vast majority of cases, it's entirely reversible. And so, for example, somebody can look like they've had a stroke, but then 3 days later, they have full function again. I've seen that kind of thing happen. So it's very different from other toxicities we've seen in the past. So it is something that's serious and relatively, I would say scary for some of the family members who see people have this happening. But overall, it is fairly well tolerated. Now, the chance of something really severe happening, like ending up in the ICU from either of these, is probably about less than 10% nowadays, now that we know how to administer these cells and how to properly treat these things earlier on. So we've gotten much better at preventing these from becoming severe. But there is still that around 10% or so risk of getting into the ICU because of this therapy, and about a 3 to 5% chance of death due to this or infections after CAR T cells, which can sometimes happen too, because your blood counts are low and your immune system is low in other ways. Your immune system's active against the cancer, but kind of low against other things. And so for all of these reasons, this isn't a simple therapy, and these side effects really need to be closely monitored at a center that has expertise and ability to do these types of therapies. And that's why you hear about it only opening up slowly in various different sites, often sites that have experience with transplantation, because we're used to these kind of more intricate toxicities, and we tend to be, most transplant centers in Canada tend to be in larger academic centers that have a lot of support, like ICU and all these other specialists to help us out as we need help. That being said, this has led to a lot of tremendous success in patients with B-cell cancers, definitely, and now in myeloma, who were deemed otherwise, palliative with no other therapies available. And this led to a lot of things in the news. You can see Steve Rosenberg on the right side there in the New York Times. He's one of the fathers of cancer immunotherapy, so to speak, talking about setting the body's serial killers loose on cancer. Not sure if that's the right message we want to be saying, serial killers, but you get the idea. And then, ASCO, the American Society of Oncology, in 2000 in 17, called this the #1 advance of the year. And, it's been covered by different places like The Economist, et cetera, and, is on Twitter and all these things. So there's a lot of hype and a lot of interest and a lot of hope really around this kind of new therapy. But really we have to talk about, where does this fit into myeloma care? And so this is where I'll hand it over to Dr. Song. I think you're still muted.
Thank you, Kevin. That was a very nice introduction. And in order to understand how CAR T cells fit into myeloma care, you have to understand myeloma to a certain extent. So I'll give you a little bit of background just so we can set the the framework for where CAR T cell fits in. So this is a slide with graphs that were taken out of this impressive article published just over 5-6 years ago, where they looked at 45,000 patients in a database in the United States. And there are 4 graphs that you can see here where people are divided by ages, 0 to 50, 51 to 65, 66 to 79, and 80 and above. And these are survival curves. And really, the way you interpret survival curves is if the curve stays higher, that means more people are alive. And if it goes down quicker, that means more people are dying. And if you look at it per era, where pink is the most modern era from 2000 to 2009, you begin to see that that curve is the highest and it separates out from the other curves. And it's very interesting because 2000, 2009 is where all the new drugs began to come out. The transplant stuff that happened in the 80s and the 90s. So this was not because of transplant, but because of all the new drugs that were happening. So you see that advance happening in the younger people, 51 to 65, and even the older people, 66 to 79, where a lot of them are not eligible for transplant. In the patients who are 80 and above, it's harder to see that separation because, of course, people who are 80 and older have other health issues. But certainly we see that it's really the modern drugs that have improved survival tremendously. And even in the databases in British Columbia, we've seen that. Next slide. So I wanted to go over some of the drugs, which I think all of you are quite familiar with. One class of drug is called the immunomodulatory drugs, and virtually, or the majority of you would have been exposed to it. The first drug was thalidomide, which was actually used for myeloma about 20 years ago, but it did have a number of side effects, for which reason the company called Celgene developed secondary and third generation drugs. Lenalidomide is the most common one, and I think a number of you would be on this drug. It's a pill, as you know. And then pomalidomide is the third generation of this drug, and this is also available to people who do have multiple myeloma. So this is one big class of drugs with some very strong potent effects and have increased survival tremendously in myeloma. Next slide. The second important class of drugs that came out over the past 20 years are the proteasome inhibitors. Now, there are 3 proteasome inhibitors that we have in Canada. One is bortezomib, otherwise known as Velcade. And the majority of you would have seen this. This is the one where you get the injection underneath the skin. And ixazomib is another drug, which is kind of like the oral Velcade. So these are pills, but they're not funded in Canada. So some of you might be getting it through some compassionate plans. And carfilzomib is the third of these drugs that are available to us. It's a bit more potent than bortezomib, and this is given intravenously. So this is the second of the classes of drugs that are extremely important. Next slide. The third class of drug that's really, really important is antibodies. Daratumumab is the antibody that is now quite prevalent, but this has only been developed over the past 5 years. And so people previously, 10 years ago, would not have had the chance to get this drug. But this drug has become extremely established in Canada now and is pretty much used with either lenalidomide or bortezomib. And we try to get everybody on this drug at some point or another in their myeloma journey. So this is the third very important class of drugs that we believe that every myeloma patient should have at some point in their journey. Next slide. But myeloma is a very interesting disease, and a disease that has a lot of positives and some that that are quite negative. One is that there are multiple opportunities to treat these patients because patients can have a chronic sequence to their disease. So their myeloma will come, they'll get a drug, they'll go into remission, then they'll get another drug, then they'll go into remission. But the problem with myeloma is that ultimately we believe that that nobody is curable. Although there is some debate about that. There are some people who I can honestly tell you who will get a transplant and who won't relapse for 10 plus years. And perhaps those people are cured, but it is really hard to say. And for the majority of people, we consider myeloma incurable, for which reason we continue to want to develop newer treatments for these patients. Next slide. So this leads into the landscape of immunotherapies, which Dr. Hay has initiated the discussion on. So if you look at a myeloma cell, it'll have all these, what we call antigen or bumps on the surface of it. And there are many of these bumps that we try to attack. One of them is CD38, and that's where daratumumab and this other drug called isatuximab, they attack CD38. So that's where the antibodies attaches to, and then eventually helps to lead to its destruction. But there is one that it's also extremely interesting and has become very important now, and that's called BCMA, and I put the arrow on BCMA. That is the bump or the antigen where there's so much focus right now in terms of the research that is being done, and particularly in the area of CAR T cells. So BCMA is attached to plasma cells primarily, although it can be found in some other cells, but primarily it's plasma cells. And so the way we have found that we can attack it is through antibodies or antibodies that are armed. So you see that more armed antibody where a poison is attached to that antibody. We have these other antibodies called bispecifics where it attaches to the BCMA, and the other side of it attaches to a T cell. And finally, we have CAR T cells that Dr. Hay has described, which is where the CAR T cell has a receptor that sticks onto the BCMA, and then the CAR T cells there expands or grows, and then it begins to attack the myeloma cells itself. Next slide. So BCMA is an important target in myeloma treatment. As I mentioned, it's expressed higher in myeloma cells than normal plasma cells. And this has essentially been one of the most important antigens that we found on the myeloma cell. Next slide. So one of the drugs that we have now, had the chance to use and that is being studied in the world is this drug called belantamab. And this is an interesting drug because once again, it is an antibody that attacks BCMA, but it has a poison. And if you see those stars, those are the poisons. So when it attaches to the plasma cells, the antibody can get internalized and the poisons are released such that the plasma cells can die, but it has other immunological mechanisms beyond that. And this is a very interesting drug and one that in the United States and in Europe is now available, but in Canada is still undergoing review. So we only have it available in clinical trials. And what is important about this drug is that it's an off-the-shelf drug, meaning that you can just get it and get it injected into you, as opposed to CAR T cells where you actually have to get T cells out of your own body, then manufacture it, and then give it to the person. So it's a multi-step process for CAR T cells. Next slide. So this goes to the study that's really, really important for all of you. So as Dr. Hay mentioned, that there are a lot of CAR T cells trials that are ongoing or that have been done, and even in multiple myeloma, they have been done as well. And they're being done by multiple pharmaceutical companies. But this is a trial that was done by a company called Celgene, which was recently bought by BMS, and that has led to the approval of CAR T cells. So when we talk about CAR T cells, in myeloma, we're not talking about one treatment. We're talking about multiple different companies making multiple different types of CAR T cells. So this is a study of this specific CAR T cell. The study is called KARMA. And the drug or the CAR T cell is, for the short form we use is ide-cel, a BCMA targeted CAR T cell therapy. This was a study that was done in many centers across the world. And unfortunately, in this study itself, there weren't any Canadian centers. Next slide. So this is a busy slide, but I just want you to focus on the cartoon itself there. And there you can see that it's a CAR T cell that's specific for BCMA. And most of the CAR T cells that are in development for myeloma right now are against BCMA, although there are other CAR T cells that are looking at other potential antigens. Next slide. So this goes back to the process Dr. Hay spoke about. In this study, first of all, you have to choose the patients that are specific for this study. So this study required patients to have had exposure to those 3 classes of drugs that I had mentioned. So if you look at the blue box on the left, you'll see that they had to be previously exposed to an IMiD agent, which is lenalidomide or pomalidomide or thalidomide, a proteasome inhibitor such as bortezomib, and an anti-CD38 antibody such as daratumumab. So patients could only go on this study if they've had exposure to all 3 of these drugs. If they didn't, they were not eligible. Then they had leukapheresis, where you took the T cells out of their body. Then it was sent to a specific manufacturing place where it was manufactured. And while it was being manufactured, the patients had to wait. They could get some bridging therapy, such as like steroids, but really the patients had to be stable enough for those couple of weeks to a month while they were waiting for those cells to be developed. And if they were well enough when the cells were developed, then they got chemotherapy, as Dr. Hay had mentioned, and then the CAR T cell infusion. And once they got the infusion, they were in the hospital for a week or 2 until they were felt to be safe, and then they were discharged. Or if they were sick enough, they got CRS or neurotoxicity, then they were kept in the hospital until they were better. And in this trial, they gave various doses of CAR T cells. So if you look on the far right, you'll see that they screened about 158 patients, but only about 140 actually got their T cells taken out. And then 128 got the CAR T cells infused. So 30 patients didn't ultimately get the CAR T cells. Next slide. So I just want to focus on just the red box. And this is to tell you the patients who were treated on this. The average person had 6 prior treatments, so meaning that they relapsed about 5 times. So anybody in the lower end, it was 3 treatments, and the higher end, somebody actually had up to 16 treatments. Just underneath that, you could see that more than 90% of patients actually had stem cell transplants. Next slide. This is the response rate, and so this is a bar graph, and this shows you how many patients actually responded to the treatment. The lowest dose was 150 times 10 to the 6th, then 300 times 10 to the 6th, and 450 times 10 to the 6th. And if you look at the highest dose, 82% responded, which means 18% didn't respond at all, and 39% had a complete response, meaning that their protein went away from their blood, whereas the others had some degree of proteins that were still left in their blood. Next slide. This is what we call the progression-free survival curve, meaning that all the people from the beginning, when they were looked at and seen as to when they relapsed or their myeloma began to come back, you could see that the curve doesn't stay flat, meaning it doesn't stay high. It just unfortunately continues to come down. So 50% of people would have progressed, meaning their myeloma has gotten worse at about 8.8 months. Whereas, there are some people that continue to do well, fortunately, but that's more like what we call the tail end of the curve. Next slide. If we remember that 18% of people actually didn't respond, and we just took the 82% of people, then we see that the curve actually looks a bit better. So 10.7 months for those who actually had a response. Next slide. But then the overall survival curve does look better. And the reason is because when people do relapse from CAR T cell therapies, sometimes they do have other treatments that they can try. And so in doing that, people actually can get to other next lines of treatments. And so the overall survival isn't too bad, with 78% of ide-cel treated patients who were event-free at 12 months, meaning that at 12 months, 78% of the people who are treated still were in some type of remission. Next slide. So once again, when you give a treatment, you also not only have to talk about how they benefited, but you also have to look at how they were harmed. And this goes to the issue of this cytokine release syndrome and the neurological toxicity. So if you look at just the box that I've put in red, that's at the highest dose where it was most effective. You see that virtually everybody had CRS, so 96%, but CRS could be just a fever. But if you look at the higher grades of CRS where people get more sick, it's only a small number, such as like 6% got higher degree CRS. The majority were just grade 1 or 2, so it was relatively manageable. Most of the CRS happened around day 1 and beyond, and it lasted for about 7 days on average. The majority of patients did require some treatment, such as tocilizumab, which is a fancy antibody to reduce the risk of these cytokines causing harm. And about a quarter of them had to require steroids to cool them off from the fevers and shortness of breath and low blood pressures that they would be getting. Next slide. The neurological toxicity that Dr. Hay had mentioned, that occurs in roughly about 20% of patients, with half of the 20% having just very mild neurological toxicity. But if you look at it, 6% had higher grades of neurological toxicity, where they were somnolent, and perhaps even worse, where they could have things like seizures. Next slide. So in conclusion, ide-cel demonstrates response in heavily pretreated, highly refractory, relapse refractory multiple myeloma. And I mentioned this sentence because I know one of the questions is, when do you use CAR T cells? So it's in patients, currently the approval is for patients who have been exposed to a lot of treatment. The overall response rate's about 73%, with a complete response rate of about 33%. Median duration of response, meaning that if you responded, was about 10 months. And progression-free survival was 8.8. And if you had a complete response, of course you did better. 19 months was your progression-free survival. And the overall survival was 19 months amongst all patients who got CAR T cells, whether you responded or not. So this trial, once again, is very important because based on this trial, the company submitted to Health Canada and got Health Canada approval to use CAR T cells in Canadians for the specific indication similar to this trial, meaning patients who have had all 3 classes of drugs. Next slide. This is a second study and the last study that I'll talk about. And I want to mention this study because this is probably going to be the second CAR T cell that will be approved, or if not, it will be the most interesting CAR T cell that we will be looking at in the near future. It's based on a trial called CARTITUDE-1, where they looked at a different CAR T cell made by another company called Janssen. So it's called cilta-cel, that's what we call it. Otherwise, if you read the whole thing, it would be ciltacabtagene autoleucel, which is a bit of a mouthful. And once again, it's against BCMA, the same antigen as before. And this trial was done in many centers, but unfortunately, there was no Canadian center that was a part of this trial. Next slide. What is interesting about this CAR T cell compared to the other CAR T cell is that when you look at the receptor, the previous CAR T cell had one specific spot on the receptor where it attached to BCMA. In this one, it's actually got 2 spots that attach. So we think that it might bind to the myeloma cells much more, easily, and so it'll be much more stickier, is what our hope is. Next slide. So once again, this is very similar in terms of the population they treated. 3 prior lines of therapy, if you look in the red box, and they had to have the 3 classes of drugs similar to the other study. Similar to the other study, once again, you screened the patients, you did the apheresis, then you gave them the bridging therapy such that you hope that over the next 2 to 3 weeks they were okay. And then finally, you gave them the CAR T cells. Next slide. So once again, they enrolled and they apheresed 113 patients. But by the time they got the CAR T cells, it was down to about 97 patients who actually got CAR T cells, because while some patients were waiting, they progressed, then they died, or they had other complications. Next slide. So going once again into the red boxes, you see that the average person had 6 lines of therapy, similar to the other study, with the person who was least exposed having 3 lines, and the most was about 18 lines. And these patients were very refractory to their treatments, which is what the other red boxes show. Next slide. This one, the interesting thing is that the cytokine release syndrome took, the median was about 7 days compared to 1 day for the other one. And virtually everybody had a cytokine release. But the majority was grade 1, which, as I said to you, is just fever alone. Grade 2 tends to be relatively easy to manage as well too. And grade 3, 4, 5, which is much more severe, tends to be a much more smaller number. Next slide. The neurotoxicity It happens in about 20% similarly if you look at the upper left box there, and grade 3 or more, just about half of them. So the neurotoxicity is similar to the other one. They also get some of these other funny toxicities, which they're trying to understand, which are not, which they're trying to see if it's different from the other neurotoxicities. So once again, they're trying to understand the side effects better and to see whether different CAR T cells can cause different side effects. Next slide. This is their response rate, which is really impressive. And if you look at the bar graph, you see that it says 96.7%. So virtually everybody responds to this CAR T cell therapy, with 67% having a complete response, and that's where the protein is not seen in their blood. So this one is very impressive. It's very similar, the study, compared to the first study, although it's probably slightly different. But when you look at just the numbers alone, the response rate being 96% compared to somewhere around 80% for the other one, this one seems to be possibly better. Next slide. And this is their progression-free survival. So if you take all the patients and you see when they progress, meaning their myeloma gets worse, here they're getting to at 12 months not reached. But if you look at the curve, it's probably somewhere around 12 to 13 months compared to about 8.8 months in the other CAR T cell. And of course, if you get a complete response, your progression-free survival is better than if you don't get a complete response. And that's what the other curve shows on the right. Next slide. So this is their conclusions. It's the side effects are manageable, that they have deep responses and their responses are very high. Next slide. So one of the reasons why I showed the second study is because they are taking that CAR T cell and they're doing CAR T cell studies in various different stages. People who are much more earlier, people who are post-transplant, they're looking at various different places where the CAR T cells might be useful. This is CARTITUDE 5, which is one of the studies that they're actually doing with this CAR T cells. They're taking about 600 patients who are newly diagnosed, where transplant is not intended. So people where transplant may actually not be possible to perform. So people who may be in their early 70s or mid 70s even, or people who are younger than that, who are unwell and who have other conditions, but who may be well enough for CAR T cells. So these patients who are newly diagnosed, they'll get a combination of drugs, which is, the Velcade or the bortezomib and lenalidomide dexamethasone for 6 cycles. After that, flip of a coin, half of them get CAR T cells and the other half don't. They get lenalidomide maintenance. So this is going to tell us, is CAR T cells in people who are borderline for stem cell transplant better than just giving them the non-transplant typical treatments? And why this is interesting for me is that we've been offered this trial in Vancouver, and this may be a potential trial that we'll do if we can get approvals from the various people that we need to. Once again, this is for newly diagnosed, so it's not going to be for anybody who has already had treatment. And so this is the interesting thing, is CAR T cell treatments are being studied at various stages. Next slide. So I want to take a step back and remind you that the CAR T cells that we've been talking about are CAR T cells against BCMA. So this is that little bump on the myeloma cells. But the issue is that there are actually other drugs that are being developed for it. Once again, like the antibody that has a poison on it and these other antibodies that we call bispecific antibodies. And these will attack the same antigen. So we may have to choose one or the other, and they'll all have different advantages. The CAR T cells are personalized, meaning you take your own cells and then you manufacture and then you give it back. And this uses the immune system. You typically require a hospital admission for that. But the benefit is that it's just one single dose. The other one, the antibody with the poison on it, that is off the shelf, meaning you don't have to wait. You can get it almost immediately. But the problem is that you have to give it repetitively, and it has other side effects. And the bispecific antibody, similarly, is off the shelf, meaning you don't have to take your own cells and manufacture. It's off the shelf, but this does require hospitalization. And of course, one of the issues is that all of these treatments cost money, with some treatments costing more than the other. Next slide. So this takes me to, I think, my second last slide, which is the issue of access to myeloma CAR T cell therapy. This is a very complicated one that I don't think I can explain very well. So this drug called Abecma, which is ide-cel, so this is the CAR T cell that I initially spoke about, the first trial. That is the CAR T cell that has been approved by Health Canada, and it was approved in May of this year. Once Health Canada approves it, Health Canada doesn't talk about who's going to pay for it. So what Health Canada does is then it pushes it to the provinces to decide whether they're going to pay for it or not. So the provinces have this organization that they all feed into called CADIT, which is the Canadian Agency for Drug and Technologies in Health. And they assess the file that the drug companies give to them, and they see what the evidence shows, and they also look at the cost of it. And unfortunately, when CADIT looked at this, they gave a recommendation of a preliminary recommendation of do not reimburse. They're trying to get feedback from other people, and they will have a final recommendation that'll come out in the next couple of months. And they decided that they shouldn't, that to recommend not to reimburse, because they thought that there was limitations in the methodology. One of the limitations is that the trial, the first trial I showed you, is that it wasn't randomized, meaning that they didn't have a comparator arm. So it was hard to see how much advantage over standard therapy you could get, but that's a hard one because what is standard therapy in this situation? The other thing that they were concerned about is the cost of this CAR T cell therapy. And the cost is at this current moment proposed to be just over $500,000. And that's just for the cells themselves. It's not for the hospitalization. It's not for all the treatments of the side effect. So this is part of the recommendations that the draft recommendations that they have. So we're waiting for what their final recommendations are, because based on the final recommendations, the provinces at that point will make a decision as to whether they will pay for this or not. Next slide. So currently, how do we access CAR T cell therapy? Of course, we want to do clinical trials, but in order to do clinical trials, as Dr. Hay mentioned, you have to be a center that can do CAR T cell therapies. And at this point in Canada, the only centers that do it are large academic university centers that do bone marrow transplants. In addition to being a center that can do it, you also have to be offered the trials. And what I mean by that is that these companies, they'll come and they may not even offer you the trial because they may not think you're the right center, or for other reasons. And so, or what'll happen is that different centers will have different trials. For instance, you may have a center in Canada that does for newly diagnosed, and another center that does relapse, and they're in different provinces. So they may have different trials, as I said. They may be an early relapse, they may be in late relapse. So, your need may not be exactly met, or the person that you're the caregiver for may not be exactly met. So the issue of clinical trials itself can be extremely confusing. Next slide. So this leads to the research that Dr. Hay is doing, because obviously we want to try to see how we can do some of this stuff in British Columbia. Kevin?
All right. Thank you for that excellent review of the myeloma new therapies that have been developed. And I just want to take you very briefly through some of the things that my lab has been focusing on in terms of myeloma research and improving CAR T cell therapy. So I divided up into really three different things. One, identifying new targets for these immunotherapies. Two, developing new immunotherapies that work better in that suppressive environment that I said happens in myeloma. And third, new ways of monitoring myeloma disease using a blood test. So I don't think I need to state this any further, because Dr. Song really covered this very nicely, but this is the KARMA trial data showing that even though, these have had tremendous improvements over, other therapies in these patients who have, basically gone through a whole whack of different therapies and have finally responded to CAR T cells. Unfortunately, the disease is still coming back in these patients. And that's what that curve is really showing us, is that out by, past a year or so, it's almost all patients have had the disease come back again. So we really need to make improvements to this therapy. And so, one thought is maybe targeting BCMA alone isn't the right approach. So my staff scientist, Daniel Waller, who's in my lab, I actually got him from a myeloma lab in McGill, and when he moved over here with his lovely wife. And basically what we're doing, what we have a grant for from the Canadian Research Society to do is to take, myeloma cells from various different patients who we've stored in that hematology cell bank. So anybody who's, donated their cells to the hematology cell bank, I just have to say thank you very much, because it helps support our research to really make things go forward. And so we take these myeloma cells and we're able to try to identify those things that are on the surface of the cells, those antigens, those bumps that Dr. Song was talking about. And we're looking at it through two different ways, one looking at the surface of the cell, one looking at, if we look at the whole cell, so we open up the whole cell and we're able to see the proteins inside the cell as well as those ones on the cell surface. And we use this fancy machine called a mass spec that you see or a mass spectrometer that you see on the far right side. And we're able to analyze all this data and really come up with, is there some certain things that are enriched on the surface of myeloma cells that aren't on normal healthy cells that would enable us to target these better. So this project is in progress currently. So the second project really that's going on in my lab is this concept that we think one of the reasons why CAR T cells might be failing in myeloma is because of that immune suppressive microenvironment that I mentioned. So if you look at this big figure here, it looks kind of complicated with all these different cells, but this TEFF cell in the middle here, this is the T effector cell. So that's the kind of cell that actually kills off the tumor cells. So in this case, we would assume that, let's say this T effector cell is actually the CAR T cell itself. Well, when it encounters the tumor cells, there's a bunch of things that the tumor cells put on their surface that say, hey, shut down, just go away and become exhausted. But there's also these macrophages, which are normally immune cells that are supposed to help the T cell response, that in this setting will start to shut down the cells. So these are called tumor-associated macrophages, or TAMs. And then you have these other cells, myeloid-derived suppressor cells, as well as T regulatory cells. So these regulatory cells, as their name implies, they regulate the other T cells there and kind of calm down the immune system. So they're helpful in the setting of preventing, autoimmune diseases. But what they end up doing in the setting of myeloma is they shut off the cells that are actually trying to kill the myeloma. So these myeloma cells can actually recruit all of these different cells to try to help shut down these responses of these T cells that are going in there. And so I have a PhD student in my lab, Lorenzo, who's working on making not just cars, but trucks. And so what we mean by a truck is basically it's a car that carries a payload. So we put a car into the T cell, but we also put into it an extra package that's able to secrete something into the environment, or it has a receptor on its surface that's able to, alter that surrounding environment locally near the myeloma cells to shut down some of the immunosuppression that's going on and turn these cells or recruit these immune cells there that have been subverted by the tumor back to the light, so to speak, back to being actual, immune cells that are trying to clear off the tumor cells. And so this is the big project that Lorenzo is working on in our lab. And we had some funding from the International Myeloma Society to have that happen. The third project, this is one that actually Dan Waller brought with him from McGill that he's been very keen to get going here, has been not so focused on myeloma CAR T cells in particular, but focused on the concept that, we need better ways of determining if we've been able to clear all of the myeloma out of somebody's body, especially when we're doing these clinical trials. And, there's basically two different ways you can detect myeloma in the body. As many of you know, there's often a bone marrow biopsy that can be done, and that's often done, at the initial stage, but maybe is not repeated because we have these other ways of monitoring the blood that are based upon looking at that protein in the blood. And the ways we typically use are what's called serum protein electrophoresis and immunofixation. And so these are good ways of detecting a large amount of protein in the blood, but actually it's not very good once it gets down to a certain level. It's hard to see the little bump that is maybe the monoclonal protein, the protein being produced by the myeloma cells, once you get down to a very low level. So you need better techniques here to look into in the blood. So to get at what we call a minimal residual disease level, or an area where it's kind of like traditionally you may not be able to see a lot of the disease activity as a protein, you need better techniques that are able to have better resolution. And so this is where we are looking into using that mass spec instrument that I mentioned to actually monitor small amounts of protein in the blood very, very accurately using this method. And the other advantage of having this method, because a lot of people would say, well, you could always just do the bone marrow biopsy, and there's lots of good methods there to find small amounts of disease. But if you look at what can happen in a patient who has myeloma, is the disease can be very patchy. And so sometimes when you stick a needle into the bone marrow, if you're not hitting the area where the myeloma cells are, you might be missing all of the disease, or you might not get a good sample, whereas in the blood, it's everywhere. And so it's more that it could actually be complementary, these two different methods. And so what you see here is that, we've talked a bit about, a complete response and a stringent response. But really what we're trying to do is get down to this point where we can get towards figuring out who has this MRD, low levels of MRD, so that we're trying to get towards where we could define what is an operational cure, so that we can say that we have really optimized a therapy. And so we're kind of working on trying to make these better detection methods, and in terms of identifying any myeloma in the body still. So in summary, the vision in our lab for some myeloma therapies that we're working on, obviously we work in a big team with a whole bunch of people, but with these projects, that our vision is to make new immune therapies against new targets that are able to work even in the immune suppressive environments, as well as make ways of monitoring patients' response to therapy better in the least invasive way possible.